So imagine you are living in a city, but this city, for some reason, has no garbage collection. It also doesn't have any storm sewers, so it kind of gets swampy and gross every time it rains. And the city, no mail delivery. Okay, honestly, I don't really want to move here. This sounds like a terrible, gross, city where nothing gets where it needs to go. And this kind of yucky wasteland is sort of what our bodies would look like if you didn't have lymphatic vessels. Think of these as little tubes that are kind of our built-in garbage trucks and storm sewers and a postal service all in one. They move this watery lymph fluid wherever it needs to go, so they're kind of a big deal. Now, the way that our bodies build and create these handy-dandy garbage sewer courier vessels, that is what Dr. Sébastien Gauvrit is studying, and he's our guest today on Researchers Under the Scope. Hey, I'm your host, Jen Cannell, and today we are looking at the way we grow new blood vessels and the way we grow lymphatic vessels, the ones moving the fluid and waste and messages around our bodies. This is where Dr. Sébastien Gauvrit comes in. Now, he's a vascular biologist and an assistant professor of anatomy, physiology, and pharmacology at the University of Saskatchewan's College of Medicine, and Dr. Sébastien Gauvrit joins us now. Bonjour, Dr. Gauvrit. Merci d'être avec nous ici à Researchers Under the Scope.
Bonjour, Jen. Merci de m'avoir invité. C'est un plaisir.
Well, and I know you were born and raised in France, and if you could take us back there for a little bit, when was it that you realized, yeah, I really like biology and science?
So I think there is two things that really trigger my interest to biology. The first one was my family. So I had the chance to spend time with my grandparents during school holidays in the countryside in Normandy. And actually, my grandfather was a gamekeeper. So, I spent a lot of time outdoors observing animals in forests and in the field. And it's also one of the reasons that I love Canada and Saskatchewan, especially for camping. And the second thing that really pushed me towards biology is a hobby that I am still doing, which is having fish tanks.
Fish tanks?
Fish tanks, yeah. So as a kid, I had a fish tank, and I had a small fish called guppies that are pretty easy to reproduce. And I actually had to understand genetics directly by mixing these different fish together to get the color or different fin shapes that I was interested in. And this really helped me to understand how physical traits are carried over across different generations. And so now it's pretty funny because...
So wait, how old were you?
So I think I was around 10 years old when I got this fish tank. And so it was pretty funny because now, and we are going to talk about that, is that I'm working with another fish, which is a zebrafish. And this, I use it for understanding vascular development.
Well, and guppies, and it's funny. Why would zebrafish, the ones you've kind of moved on to, what makes them useful for you as a biologist?
So zebrafish is a fantastic model to visualize things. So including vascular biology, because they are, during development, their embryos are transparent. So this allows you to really visualize the formation of blood and lymphatic vessels at the cellular level. And so you can understand directly how these cells migrate and how they invade specific organ, like the brain or the trunk, to form these new blood vessels. One fact that is interesting in comparison with human species is that we share around 70% of our gene with this species, with zebrafish. And so actually most of the genes that trigger vascular disease in human are present in the fish.
Okay.
And using new molecular techniques such as CRISPR-Cas9, you can directly target a specific gene to block its function, or you can make the cells that make blood and lymphatic vessels fluorescent, and so you can visualize everything in fish.
Well, I remember meeting the zebrafish at the U of S because of Michelle Collins. they're really cool. And when did you start working with them, like to try breeding them and to try to see what traits were coming out?
The zebrafish, I used them during my postdoc when I was in Germany. So I was in the laboratory of Didier Stenier, who was one of the pioneers in the field of the zebrafish model. And it was a really fantastic experience because we had access to a lot of different genetic transgenic lines, and so we could visualize everything as well. Like we had a lot of microscopes that really allow us to kind of go inside these pathways.
Well, and you mentioned that these are transparent fish. Like you can actually see them while they're still alive under a microscope?
Yeah, so it's really amazing. So for the first few days, and even later on, you have fish that do not develop pigment, and so you can see all across their body, so you can visualize full organ and not only organs, but also the vessels that colonize these organs. And these fish have also been very, very useful in terms of human genetic disease, especially in the lymphatic field. So, for example, there is a specific disease called the Hennekam syndrome, where it was affecting a few patients. And so these patients have a specific lymphatic vessel defect. And actually, the gene that's responsible for this disease was identified in zebrafish first during a genetic screen in Netherlands.
Wow. And so zebrafish have been very useful for a lot of like the cardiovascular and just figuring out how we develop. And you said we share 70% of our DNA with them?
Yeah, 70% of our gene, not our DNA or genes. So it's more the specific sequence. But in terms of function, there is a lot of redundancy. This concept across evolution, right? If you know how to make a blood vessels as a fish, your evolution are going to keep the same mechanistic to make it work in mammals, and including us.
I kind of want to know, okay, for mammals then, how do you translate this? Like, and do you have any other animal models that you're working with?
Yeah, so I'm also using the mouse model to investigate the question we have in terms of vascular development. And so mouse are, as you know, a very great mammalian model. And it's thanks to this model, you have access to a different set of genetic tools. And we are using this specific set of genetic tools to complement our research with zebrafish to kind of understanding vascular development. And people are still, which are the best model, but at the end, it's what model will bring you the best answer of your question. And so that's why I was trying to highlight is that it's complementary and not which one is the best. It's more which question you have in mind and what tool you can have in each of the organisms that can bring you the answer.
Yeah. Yeah, and then trying out other things and trying them with different models. That's just really cool. Mm-hmm. What questions is your lab trying to answer right now?
Right now, we are trying to understand how to make blood and lymphatic vessels. And more specifically, what are the genetic players that make a cell choose between being a blood vessels or being a lymphatic vessels? So how these cells become one or the others or both? And so we have identified specific genes that might be involved in this kind of decision between these two cell types. And so we are trying to understand how does it work using fish and mouse.
What are the genes you identified? What are their names?
So this gene that regulate the kind of a specification or maybe the cell choice, cell fate decision is called HX for hematopoietically expressed homeobox. This is a transcription factor. That means it's a gene that is going to be expressed in the nucleus and it's going to trigger the activity of other genes. So it's kind of a cascade of signaling that will be triggered by this gene. And so if you understand how this gene regulates others, we can identify new genes involved in lymphatic disease and understand a bit more the process behind all these events.
And what are a few examples of lymphatic disease?
One example that came to mind is actually was a great story using the zebrafish to understand the lymphatic disease. And so it was also a case for using zebrafish as a tool for personalized medicine, which means that you're going to tailor the treatment to the specific patient. And so a few years ago, it was a case in the literature where it was a 12 years old boy that was carrying a mutation in a gene, which trigger an overgrowth of the lymphatic. And so you have too much lymphatic in your body, and notably in the lung. And so this was impeding his function of breathing, which was life-threatening, and he was not responsive to regular treatment. And so what they did, so the researcher that was taking care of him find the mutation using genetic sequencing. And what they did is they used the zebrafish to speed up the ability to test drugs to block the growth of lymphatics. And so they introduced the mutation of the gene inside the lymphatic system of the zebrafish. And they use a fish that carrying the same mutation as the patient to screen a lot of different drugs. And they actually find one drug that could rescue the fish by reducing his lymphatic overgrowth. And they actually move very quickly to the patient because it was life-threatening situation. And actually the patient recovered from his condition thanks to the drug that was found initially in the zebrafish.
Wow. And so if I get this right, they figured out where the mutation was in the boy. They created the same mutation in a group of zebrafish. And then they tried throwing different drugs at these poor zebrafish, trying to figure out, okay, what are they going to respond to? And when they found a really good match, they tried it on the human, and it did work.
Yeah, that's right, yeah.
That's incredible.
It's really, really exciting, yeah.
Oh, this is going to sound dumb, but how do you make the zebrafish take drugs?
So it's relatively easy because, as you know, they're in the water, so they're swimming around. And so most of the drugs can be diluted in specific solvent, and then introduce this solvent containing the drugs inside the water. And then the fish actually will take these drugs contained in the water pretty easily. And so that's why also fish is not only a great model for genetic, but also for toxicology, because it's easily take out from its environment a lot of chemicals. And so it's great to understand also how the environment trigger specific diseases. And in this case, it's really great to use it as a drug screening purpose.
Ah, so you don't have to inject them. You just literally put it in the water that they swim in.
Exactly. It's very practical.
The mice you would have to inject.
That's right. Yeah.
Okay. Yeah, a little more detailed, but yeah, mammals are a little more detailed. With the University of Saskatchewan, you came on as an assistant professor last year. I know you've been getting your lab set up, and I understand that last week, we're in mid-July at this point, you got a really cool new microscope. Can you tell us about what your lab setup is like and what you're trying to do in there?
Yeah, no, so it's a very, very exciting news, and we are very excited to use this tool that we obtained thanks to the NSERC funding. So again, I'd like to thank NSERC for the ability to do our work. And so thanks also to this grant, we will also be able to expand the fish room at the university. So this will provide us more space for not only me, but also for other researchers that work in different aspects of developmental defect, such as a craniofacial defect also. We have two of my colleagues, Dr. Botner and Dr. Zabo Rogers, are also interested to use this model. And this microscope will give us the opportunity to image our fluorescent fish and characterize better the new genetic tools we are generating at the moment. So at the moment, I have 3 and now soon 4 people in my lab. So I have 2 summer students that are doing a great job at the moment, Cole and Zaina. I have also a research technician with me, Luba. Is also doing a fantastic work. And I'm going to have also a master student starting in September, Renam, that will join us, and we're going to start to do great things. And so this microscope that we get, we are excited to use it because it also has the ability to improve the image quality because it has a specific image processing algorithm that is provided by a specific software. And so this really improves our ability to visualize this event occurring during vascular development.
Oh, that is cool. And I know I asked you about the questions that your lab is trying to answer, and I know we talked about HX, but are there other things you're exploring as well?
Yeah, that's right. So we have we are looking at we are also working on the main growth factor of blood vessels. which help them survive and grow. And so this name is VEGFA, so vascular endothelial growth factor. So this is the main growth factor that makes this cell survive and proliferate. And it's implicated in a lot, if not all, human vascular disease. One specific example is an ophthalmic condition that is called AMD, for age-related macular degeneration. And so this AMD blur your central vision. And so this is due to an overproliferation of blood vessels in your eye. And so one of the problem is that patients are treated with an injection that blocks this growth factor. But one problem is that it requires multiple injections in the eye, which is not great, as you can guess.
Ooh.
And a high proportion of patients, they are going to develop a resistance against this therapy, which is also a big issue.
Oh.
Yeah. It's also a very costly therapy for the country, for the healthcare. So it was estimated that these therapies cost more than $500 million per year.
Per year?
Per year. This was in 2017, so you can guess that now we're in 2025, so the cost must have way high up. It's also an age condition, so you can guess with the aging of the population, this issue will become even more, will become stronger. So that's also one of the interests for us to look at these pathways. So we are looking at this gene in zebrafish because it's conserved, it's present in the zebrafish as well. And we try to understand basically how it works, how does it acquire this resistance and how you can understand more this process. And on the long-term goal, we want to try to find a way to reduce the resistance to the therapy or find new forms of treatment to block the growth of blood vessels.
Yeah, because if the only treatment right now is like multiple injections in the eye and it's incredibly costly, like $500 million to try to run this, you're going to have a lot of people who can't afford that or who are in areas where it's just not available and they lose their sight, right?
Yeah, that's right.
Oh, so how do you investigate, like how do you actually take apart that and kind of look at what it's doing? Like what in the lab has to happen for you to understand what's going on?
So we are relying on a genetic tool. So we have mutation in this gene for both HX and VEGFA. And so we basically remove the gene in this fish and we try to understand how this fish recover from this. In the case of VEGFA, basically we remove the gene. The fish can be rescued. That means they can grow without the gene, which is very unique because in mammal, for example, if you compare that to mouse, the mouse model die very, very early during development versus the fish because it evolved in water and has access to more oxygen because of the diffusion of oxygen inside the tissue, can survive way longer compared to a mammalian model. And so it's one of the advantages of the zebrafish compared to the mice for this gene is that you can investigate questions way later in development without too much affecting the system. And so that's why we are trying to understand how this fish make blood vessels without growth factor. How do they cope for the lack of this and try to understand this in a comparison to the human model where patients develop these new blood vessels as well.
Oh, well, this is really cool. And I know this is thanks to those 2 awards you managed to win from NSERC this year. Congratulations. That is a big deal.
Thank you. Yeah, it was. We're very excited about that. Yeah.
Into the future, and I know you've had to explain this to so many people, but in the future, if you can pinpoint a few answers to even some of these questions, what diseases can you see this helping us treat?
So I think what I like to say is that for me, it has a broader perspective because the vasculature is present in all organs. And so this could help to a wide range of disease from lymphedema to stroke. And this disease I want to mention is macular degeneration. And what is also exciting in the field is that even now, and I think this is something that I'd like to tell people, is that we still discover new things. It's not like we have a textbook and we know everything. One example I really want to highlight is that the lymphatic system, for example, for the lymphatic system has been discovered in the brain a few years ago. But for a few hundred years, people believed that there was no lymphatic system in the brain. And so this kind of new discovery, discovering a new vascular system in an organ that was very well known, that you think was known for hundreds of years, really trigger opening a lot of questions about how these lymphatic vessels and their relationship, what they are doing in the brain, and what they're all doing in neurological disease. Because, for example, lymphatic vessels also carry immune cells, and so there is a lot of not only removing the waste of the brain, but also bringing immune cells into the brain, and how this is affected in neurological disease really open a lot of questions.
And in the brain, where do you find the lymphatic vessels? Like deep in the middle or at the outside? Where do they live?
So that's one of the reasons that people had some issue to find them, is that they are not inside the brain per se. They are on the surface of the brain and kind of just under the scalp. That's why they were hard to find. And actually, there is hypothesis saying that maybe these lymphatic vessels are important also during sleep, because, the fact that we sleep is to remove the waste of the brain. So you have different circulatory system in the brain, but maybe these lymphatic system are more activated during the night and kind of remove the waste from the brain. And so this really, again, opened a lot of questions about in the field.
They're really neat little cells. The little garbage collectors, our sewage guys. They bring the parcels, the mail in the form of like white blood cells. They're really neat.
Yeah, definitely. It really, I think, feeds the imagination of people. I mean, like for example, the Hennekam syndrome, when the people also found the gene responsible for this disease in fish. I read that when I was doing my PhD, and I really oh, this is what we have to do. I have to do that because it's awesome. By pure luck. There's also one thing that nowadays researcher has to answer questions and know exactly what they're going to find. And the old genetic screen that was provided in fish were kind of random, because the way it was working is that you take a bunch of males, you treat them with a chemical that induce random mutation in different genes, and then you're going to breed them with females, and then you have families that carry random mutation. You can cross them to identify any defect, growth defect or organ defect. And one of these random genetic screen was done looking at blood vessels. And this is where they find these fish that had no lymphatic vessels. And then they find out that it was linked to this human condition. And so it's interesting to see that just by serendipity and by randomness, sometimes you can find a bit greater science than when you have a very specific questions in mind, but it's another topic. It's an interesting concept. Yeah.
That's that sense of serendipity, though. I think science would be so boring if you didn't have that.
This kind of approach was actually a Nobel Prize, not for zebrafish, but for the Drosophila. That was one of the first genetic screen. But this kind of techniques or approach really was already there and was rewarded. So it's pretty interesting.
You mentioned that, this has been an interest of yours, like the fish part, since you had those guppies and you were maybe 10, 11 years old, and you still have fish at home now? Fish at work and fish at home?
Yeah, I still have a fish at home. Actually, still guppies. I'm still playing with it. I keep this. I always had a fish tank in every country I went. Either in France, Germany, or here, I always had a fish tank at home.
Well, I'm really glad that you were one of our fish guys, and I wish you luck with this. Congratulations on everything, and thank you for being here on Researchers Under the Scope.
No, thank you. It was great.
Dr. Sébastien Gauvrit is a vascular biologist who studies the way we form new blood vessels and lymphatic vessels. His research could change the way we treat everything from lymphedema to strokes, even to macular degeneration. This year, Dr. Gauvrit won 2 major grants from the Natural Sciences and Engineering Research Council of Canada, NSERC, and he's an assistant professor of anatomy, physiology, and pharmacology at the University of Saskatchewan's College of Medicine. Researchers Under the Scope is a presentation of the Office of the Vice Dean of Research at the U of S College of Medicine. We record and we produce this podcast on Treaty 6 territory, and we pay our respect to the First Nations and Métis ancestors of this place. We reaffirm our relationship with one another, and we'd sure like to thank you for sharing your ears with us, for listening to the show and being a part of research. I'm your host, Jen Cannell. Thanks for listening to Researchers Under the Scope.